Conquer lipid classification for your next competitive exams. This video offers simplified approach to understanding different lipid categories. We will dive into importance of each class of lipid and equip you to ace those tricky MCQs. Master lipids, master your exam.
Lipids are different from all other biomolecules. Carbohydrates are polymer of monosaccharides. Proteins are polymer of amino acids. Nucleic acids are polymer of nucleotides. Lipids, unlike all these biomolecules, are not true polymers. They are relatively smaller in size and, most importantly, non-polar, means insoluble in water. Lipids can be defined as they are heterogeneous substances grouped together under one class based on their common property of being insoluble in water and soluble in non-polar organic solvents like ether, benzene, chloroform, hot alcohol, acetone, etc. Or you can define as compounds which are relatively insoluble in water but freely soluble in non-polar organic solvents like ether, benzene and so on. Now let's see the classification. It might seem complex at first, but by the end of this session, you will have a clear understanding and be able to answer those related MCQs. We will break it down and make it easy to remember. Classification of lipids was proposed by Bloor in 1925. So it's called Bloor's classification of lipids. Lipids are divided mainly into three parts: simple lipids, compound lipids and derived lipids. Simple lipids are made up of fatty acid and alcohol, mainly glycerol. Compound lipids are made up of fatty acid, alcohol and additional group other than fatty acid and alcohol. And derived lipids means products obtained from simple lipids, compound lipids or precursor of these lipids. For example, fatty acids, glycerol, cholesterol, vitamin D, steroids, etc. I have created a video explaining fatty acids and their classifications. Click on card in right corner and you can watch it. First we will see simple lipids. They are of two types, oils and fats, made up of fatty acids and alcohol, mainly glycerol. It is called triacyl glycerol because 3 fatty acids and 1 glycerol. I will make whole dedicated video on triacyl glycerol and their properties. Second is waxes. They are ester of higher fatty acids with monohydroxyaliphatic alcohol but not glycerol. Waxes are used as the base for preparation of cosmetics, ointments, polishes, lubricants and candles. Next is compound lipids. Don't be scared of this large image. Just clear your funda. Compound lipids are fatty acid plus alcohol plus additional group. Now if additional group is phosphate, it's called phospholipids. And if phosphate is not there, it is called non-phospholipids. First we will discuss about all phospholipids. They are classified based on the alcohol compound. If the alcohol is glycerol, it is called glycerophospholipids. And if alcohol is sphingosine, it is called sphingophospholipids. Now see the structure. It is phosphatidic acid. It is made up of one molecule of glycerol where hydroxyl group of first and second carbon atom of glycerol are linked to fatty acids and third hydroxyl group is linked to phosphoric acid. Now if phosphatidic acid binds with nitrogen, it is called nitrogen containing glycerophospholipids. If the nitrogen base is choline, it is called phosphatidyl choline, lecithin. If it is ethanolamine, then it is called phosphatidyl ethanolamine, means cephalene. If nitrogenous base is serine, it is called phosphatidyl serine. And next is plasmalogens that we will see later on. Phospholipids in general are amphipathic, particularly lecithin. They have both hydrophobic and hydrophilic portion in their molecule. The glycerol along with phosphoric acid and choline constitute the polar head of phospholipid molecule, whereas hydrocarbon chain of fatty acids represent the non-polar tail. When phospholipids are mixed with water, the polar heads orient themselves toward aqueous phase while non-polar tails orient towards opposite direction, forming molecular aggregates called micelle. Right side image is of liposome. Liposomes are microscopic spherical particles prepared by sonification of mixture of phospholipids and cholesterol. Phospholipids form bilayer enclosing some water. Drugs, proteins, enzymes, genes, etc. may be encapsulated by the liposomes, which could act as a carrier for these substances to target organs. They exhibit superior pharmacological properties also. Liposomes can be used in cancer chemotherapy, antimicrobial therapy, delivery of vaccines, and diagnostic imaging. The constituents of lung surfactants are dipalmitoyl lecithin, phosphatidyl glycerol, cholesterol, and surfactant proteins A, B, and C. In dipalmitoyl lecithin, fatty acids attached to the first and second carbon of glycerol are palmitic acid. It is secreted by pulmonary type 2 epithelial cells. Its main function is to reduce the surface tension at air-liquid interface in the lung and prevent the collapse of lung alveoli. During fetal life, the lung synthesizes sphingomyelin before 28th week of gestation. But as the fetus matures, more lecithin is synthesized. The lecithin to sphingomyelin ratio should be more than 2 for fetal lung maturity. Lecithin to sphingomyelin ratio in amniotic fluid is an indicator frequently used to evaluate fetal lung maturity. Prior to 34 weeks of gestation, concentration of lecithin and sphingomyelin are equal. Later, the concentration of lecithin rises markedly and lecithin-sphingomyelin ratio becomes 5 at term. Lecithin-sphingomyelin ratio less than 1 resulting in respiratory distress. Respiratory distress syndrome is one of the most common causes of morbidity in preterm neonates. Patients present shortly after birth with apnea, cyanosis, grunting, inspiratory stridor, nasal flaring, poor feeding, and tachypnea. Radiological findings include a diffuse ground glass appearance. The preterm infant who has RDS has low amount of surfactant. It is due to the defect in biosynthesis of dipalmitoyl lecithin that causes the collapse of alveoli. Postnatal surfactant replacement therapy significantly reduces the incidence and severity and morbidity. In pulmonary alveolar proteinosis, alveoli fill with proteinaceous materials. Congenital Pulmonary alveolar proteinosis is caused by inborn errors of surfactant protein metabolism. The prognosis for infants with congenital PAP has been unfortunately poor and they die within the first year of life. After phosphatidyl choline, next is phosphatidyl ethanolamine. It is also called cephalene. Here the nitrogenous base is ethanolamine. They are found in biological membrane and possesses amphipathic properties. They have role in blood coagulation. They accelerate the blood clotting as they share the structure of thromboplastin, which is essential for blood clotting. Next nitrogen containing phospholipid is phosphatidyl serine. Here serine is a nitrogenous base. They are component of cell membrane and involved in memory functions and apoptosis. Next is plasmalogens. When fatty acid is attached to C1 of glycerol in glycerophospholipids by an ether linkage instead of usual ester linkage, they are called ether lipids. For example, plasmalogen. On C1 of glycerol, unsaturated alkyl is attached by an ether linkage. Plasmalogens occur in brain and heart. It may have protective effect against reactive oxygen species. It is also an precursor for platelet activating factor, where at C1 alkyl residue is there and at C2 ester linkage is with acetyl group. Platelet activating factor activates blood platelets, causing them to aggregate. Here we have finished the nitrogen containing glycerophospholipids. Now we'll see non-nitrogen containing glycerophospholipids. Non-nitrogen containing glycerophospholipids contain glycerol, fatty acid, phosphate and non-nitrogenous compound. For example, phosphatidyl inositol, where inositol is attached to the phosphatidic acid. Second is phosphatidyl glycerol. And third is diphosphatidyl glycerol, or you can say cardiolipin. We will see one by one. Phosphatidyl inositol is found in brain tissue and plays important role in lipid signaling and cell signaling. The inositol ring can be phosphorylated by a variety of kinases to form phosphatidyl inositol 4,5-bisphosphate. Actions of many hormones are mediated by phosphatidyl inositol. Hormones stimulate phospholipase C to hydrolyze phosphatidyl inositol 4,5-bisphosphate to form inositol triphosphate and diacylglycerol, which acts as second messenger for hormone action. Next is phosphatidyl glycerol. You know that it is a component of lung surfactant as we have discussed earlier. Next is diphosphatidyl glycerol, or you can say cardiolipin. Diphosphatidyl glycerol is where two phosphatidic acids are esterified to one glycerol by phosphate groups. It is also known as dimeric phospholipid. It makes up 15 to 20% of inner mitochondrial membrane and is crucial for both mitochondrial function and cellular processes outside the mitochondria. Impairment of cardiolipin synthesis plays a role in mitochondrial dysfunction. Mitochondrial dysfunction is associated with pathogenesis of diabetic cardiomyopathy, myonecrosis, stroke, peripheral vascular disease, nephropathy, retinopathy, and encephalopathy. Cardiolipin exhibits antigenic properties. Antiphospholipid syndrome is an autoimmune disease where anti-cardiolipin antibodies are formed. Detection of ACA is of diagnostic importance in the detection of syphilis, idiopathic spontaneous abortion, and systemic lupus erythematosus. So that is all about glycerophospholipids. Now, sphingophospholipids. Sphingophospholipids contain sphingosine, an amino alcohol instead of glycerol, along with fatty acid, phosphoric acid, and choline as nitrogenous base. For example, sphingomyelin. As the name indicates, it is rich in myelin, the sheath surrounding the axon of neurons which functions as an insulator. Now we will see non-phospholipids. Glycosphingolipids, or you can say glycolipids, sulpholipids, and lipoproteins. Glycosphingolipids or glycolipids are made up of fatty acids plus sphingosine as alcohol and carbohydrates. Cerebrosides have monosaccharide glucose or galactose. It is present in brain white matter and myelin sheaths. Globosides have oligosaccharides. They are present in erythrocytes membrane. They are essential structure of blood group P antigen. Gangliosides have oligosaccharide and in addition sialic acid, which is NANA, N-acetyl-neuraminic acid. They are present in grey matter of brain. Monogangliosides means having one sialic acid are of three types: GM1, GM2 and GM3. They are widely distributed in body tissue and acts as receptors. GM1 is inhibited by cholera toxin. Sulpholipids are glycolipids containing sulfate group. They are present mainly in nervous tissue, brain and nerves. They act as electric insulators of nerve impulse. Lipoproteins include chylomicrons, VLDL, LDL, and HDL. So finally we are revising. Lipids are simple lipids, compound lipids, derived lipids. Simple lipids, fatty acid and alcohol, mainly glycerol, oils, fat and waxes. Compound lipids, fatty acid, alcohol and additional group. If it is phosphate, then phospholipids. Phospholipids again depending on the alcohol, glycerophospholipids, and if it is sphingosine, that is sphingophospholipids. For example, sphingomyelin. Glycerophospholipids again if it is nitrogen containing base, phosphatidyl choline, which is important in lung surfactant, phosphatidyl ethanolamine, which is important for blood clotting because of similar structure to thromboplastin, phosphatidyl serine, which is important for memory function and apoptosis, and Plasmalogens, which is important precursor for platelet activating factor. If glycerophospholipids having non-nitrogenous containing compound, Phosphatidyl Inositol, which is important for hormone action, Phosphatidyl Glycerol, lung surfactant, Cardiolipin, which is important for mitochondria, and also check out for antiphospholipid syndrome. Now if additional group is not phosphate, then non-phospholipids. But here alcohol is sphingosine. First is Glycosphingolipids, cerebroside, globoside and ganglioside. If glycolipid contains sulfate group, then sulpholipids. Lipoproteins include chylomicron, VLDL, LDL, HDL. Derived lipids include fatty acid, glycerol, cholesterol, vitamin D and steroid. This covers the topic of lipid classification. If you find this video helpful, let me know in the comment below. Feel free to share this information with others who might be interested. Press like button and subscribe my channel. Thank you.